You're listening to the Future Tech Podcast with Richard Jacobs. Future technologies such as artificial intelligence, stem cells, 3D printing, gene editing, Bitcoin, blockchain, the microbiome, quantum computing, virtual reality, and exploring space are much closer than you might think. In fact, many early versions of these technologies are in play right now, and the companies that are using these technologies are the focus of this podcast. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a thorny medical problem. Remember, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you've enjoyed the podcast, please listen, subscribe, like, and tell your friends about it. Thank you. Hello, this is Richard Jacobs with the Future Tech and Future Tech Health podcast. I have Monica M. LaRonda. She's an assistant professor of pediatrics and endocrinology at Northwestern. We've been talking about a bioprosthetic ovary that was created using the 3D printing method, which she'll get into. So, Monica, thanks for coming.
Yeah, thanks for having me. I'm always excited to talk about our research.
Yeah, maybe it's obvious, but what's the reason to try to recreate or 3D print an ovary?
Yeah, our main patient population are childhood cancer survivors. So those children that either have a disease or treatment for their disease that would render them infertile later in life. And so they would have reduced reproductive capacity and reduced sex hormone function. And so developing a bioprosthetic ovary or a 3D printed ovary is one way that we're trying to advance their regeneration options.
Huh. So, how far along in this process are you? Have you been able to construct an ovary and did it have any functionality at all, or is it still really early on in the process?
Yeah, we've been successful in mice. So we've been able to 3D print a scaffold that supports the ovarian cells that are important for function. So the potential egg cells or the oocytes and the surrounding supportive hormone producing cells. Together that's called an ovarian follicle. And it's critical. One of the things that we discovered was that it was very critical to have this spheroid structure, this cell aggregate structure maintained in a particular 3D printed geometry that would allow them to grow and mature and ovulate. First, we found that it ovulated in culture, and then we were very excited to then transplant these bioprosthetic ovaries in mice whose ovaries were removed, mate them, and they were able to produce pups. So their hormone and fertility was restored with this transplant.
Oh wow! How I haven't really thought about ovaries very much. I don't think most people have, but
no.
in a normal ovary, I would guess you'd have the cells dividing normally, and then how is the actual egg cell produced? Is it a differentiation of a different cell type that exists in the ovary, and that becomes an egg cell, or how does an egg cell come into being?
Yeah, that's a really good point, and it brings me to a very important factor about female fertility, which is that our oocytes are a finite resource. They're actually, once we're born, we have the most number of potential egg cells that we'll ever have, and it just declines from there until natural menopause, or until something destructive comes along, either a genetic mutation or a disease or a treatment that would affect those cell numbers. The oocytes are actually paused in meiosis, which, if you remember from biology class, is not the same as mitosis. So they're not doubling. They're not copying themselves. They're trying to create half their genetic material so that the two halves can be combined and develop into an embryo and a new human being. The two halves being the egg and the sperm. So they're frozen that way at birth.
Okay, so from what you've observed, it is true that by the time an organism is born, it has all the egg cells it's going to have, and then they hang out there not doing much, and then according to some hidden timing schedule, one or more of them, every month or every gestation cycle will now suddenly undergo meiosis and create what?
Yeah, so I think the egg expert colleagues that I have would want me to point out that the egg is not just hanging out there doing nothing, that is actually an important contributor to the process. But in some sense, it's paused in meiosis, but it does really grow and expand, and that's one of the aspects that we have to consider when we create the 3D printed scaffolding, that the oocyte itself and the cells surround it will expand to over 600 times their original size. And so you just have to have this dynamic structure that is able to hold them. And so as it's growing, it kind of prepares itself, and it's kind of a black box as to actually what happens in some respects, which makes doing this sort of research really fascinating. But it readies itself to complete meiosis upon ovulation, and then the sperm will combine its half of the DNA to then first create a zygote, and then develop into an embryo. If that implants, then it becomes a fetus and a whole unique being.
Right, but even considering, I'm realizing how little I know. So when you 3D print an ovary, you said you've done it in mice. I mean, are you 3D printing? I guess you're 3D printing egg cells as well, right? But are they? I mean, so when a person's born, are their egg cells immature and they age with the person, and then when the person reaches a certain age, then the cells are ready according to some clock to start being candidates for meiosis and traveling down the fallopian tubes? I mean, or are these egg cells like ready at all times to be harvested?
Yeah, that's a great question. And I think what might help is if I create that link between the patient and how we're going to use like this regenerative medicine type of technology. So when a child is diagnosed with cancer, and they're going to receive this treatment that puts them at over 80% risk of developing infertility, they qualify for our ovarian tissue cryopreservation procedures that we have at Lurie Children's Hospital. I co-direct the fertility and hormone preservation and restoration program with my colleague, Erin Rowell, who's a pediatric surgeon. And her and I offer this clinical procedure to patients. And so one ovary is removed ideally prior to their initial chemotherapy or treatment. So before it would be exposed to these potential harmful effects. It's processed and then cryopreserved. So there's been several patients who have had offspring, so have gotten pregnant and had live births from ovarian tissue that was transplanted after it had been cryopreserved in this same way that we do here in the clinic. However, there's many cancer patients that have metastatic disease, and so they wouldn't qualify for this transplantation procedure because we don't want to give them back the disease that they just survived. And so what we do with the tissue is we isolate out the cells that we want. So we want the oocytes from that patient because it will have their biological material, which is very important for them to pass on to their offspring. And we'll also harvest the hormone cells, and we'll put that into a 3D printed scaffold that we make in the lab. And so then the transplantation happens like it normally would have, but this time we know that there's no metastatic cells in there, and hopefully we can even make a better version of what exists with the tissue today. So the scaffolding itself is what's actually printed, and it's at a specific architecture to hold those ovarian follicles, which is the potential egg cells and the hormone producing cells. And we seed those into the scaffolding.
I got that, but I guess my question was, if I have someone that's 5 years old,
Mm-hmm.
and I cryopreserve one of their ovaries, and now they're 25, everything looks good, and I put the ovary back into them, they can have kids within what time period? Pretty quickly?
Yeah, that's a good question. So most of the live births that have happened are from adults or post-pubertal children that have been cryopreserved when they were post-pubertal and then transplanted back when they were adults and wanted to get pregnant. So there's only been 2 cases where this tissue was transplanted back that was preserved prior to them going through their full pubertal transition. So we hope that it works the same. We know from animal studies that and from some human studies that we can get eggs produced from these immature primordial follicles that are collected from pediatric patients. But we're actually doing a large research study to figure out the quality and the rate at which this can happen, because you're right, it's coming out of a person who doesn't have that hormonal cycle, which is what activates those follicles and matures those oocytes into good quality eggs. That happens through puberty and after puberty in an adult woman.
Yeah, I wonder if anyone's done a longitudinal study on oocytes and looked at them, over a person's lifetime. Because like, for instance, a woman's cycle has definite timing to it.
Right.
So you wonder if the first, if a woman's first cycle matures all her eggs to a point where they're candidates to, to travel in the fallopian tube, or if a given egg takes X number of cycles.
No. There's a few oocytes that are recruited at a time, and then one very special one is ovulated. And that happens, between 28 and 32 days in a normal cycling female. But not all of them are all recruited. Yeah.
Oh, so once you're chosen, you're picked out of the bunch. That's it. It's either go or no go. And what happens?
Yeah,
to
it's either die or they die off, or they're ovulated. But it's a cluster of them at a time. But it depletes. Like we're born with about 300,000, and then it depletes down to about 1,000 per ovary once we reach menopause. And so, there's several recruited that don't make it to eggs, but not all of them are recruited at once. It happens over decades.
Well, okay, I gotcha. But again, no one's done a study on the ones that are just hanging out in the freezer, essentially, or hanging out in the ovary, not matured yet or not picked, and what happens to them over time.
Well, there's been some studies definitely of because we do the way that we do assisted reproductive technologies with like IVF, in vitro fertilization. Many families have to go through that process where the woman has an increased hormone regimen to recruit more ovarian follicles to grow, and then we're capturing them before they get ovulated out of the ovary, and then using those in the lab to make embryos, once combined with sperm, or to freeze them for later. And so we do use some of those eggs that wouldn't normally be ovulated to create embryos and to create beings with the assisted reproductive, with IVF and things like that. But we are studying, I think where there's a lack of research is the prepubertal and through the pubertal transition, which I think because I'm in the Department of Pediatrics, I'm very interested in, like what happens during that transition, and how does the ovary change, and how do the follicles' capacity to be good quality eggs change over time.
Can you tell which eggs are going to be next on deck to be developed? Like the way the eggs are they stored in like a regular like honeycomb pattern? And let's say the ones on the quote unquote top or nearest the exit are the ones that are most likely to be, taken for the next batch. Is there any?
There's a lot of theories out there. So the ones that are kind of the bank, we call them the bank of eggs or potential eggs. Those primordial follicles, they're in a more rigid part of the ovary. So they're in what's called the cortical region. And then they migrate into the middle of the ovary. before they expand and then reach the edge again where they then ovulate out. But the growing happens within the middle, which is really interesting. And the bank stays on the outer perimeter. So there's a lot of theories as to which ones are going to be recruited next based on, which ones were formed first in the embryo, which ones underwent meiosis first or completed the first part of meiosis to be, then quiescent. And then also where they were located in the tissue, not just in the cortex, but were they next closer to the kidney or further away, for example. There's there's theories out there, but I don't know that answer.
Yeah, I'm sorry I'm asking this stuff that's coming to mind, but I just realized, this is something that's very complex, and I haven't really thought about it at all. Most people haven't. So that's why I'm asking these questions, because all this stuff's popping into my head now, and I'm like, wait a minute. So
yeah, and this is why using 3D printing is interesting and I think useful in this particular case, because the ovary is complex and compartmentalized, and we're publishing a study soon from my lab where we investigated all of the structural proteins of the ovary and mapped it spatially throughout the ovary to define these not just rigidity differences, but biochemical differences that might actually affect how follicles behave in an attempt to make the best possible ovarian bioprosthetic.
So this is not necessarily a permanent solution, but is this maybe a short-term one where, like you said, someone's had childhood cancer, you were able to preserve at least, the egg cells, if not the whole ovary itself. And now is your best hope to give them a window, like a 3 to 5 year window or something, where they could have natural born children, but that's about all you're going to get? Or like, what's the goal of the project?
Yeah, that's a great question. So when we transplant back ovarian tissue, the average age that tissue lasts, and so it's quite often about 20% of the tissue is put back at a time. It lasts about 2 to 5 years, but sometimes it lasts longer. Sometimes it lasts like 12 years. And this is based on, the type of ovarian sex hormones that are produced and testing that within the blood. And so our goal is to have a long-term transplant, and we might be able to do better than the natural ovarian tissue that exists in those type of transplants, just because we might be able to control the environment a little more with the scaffolding, with the 3D printing. And so we're hoping to get a long-term transplant, but I think short-term restoration of fertility is definitely one of our key targets when we first start moving these into the clinic. First looking for hormonal restoration, and then seeing if we can get some short-term year or two ovulation to help them have a biological child.
I figured, yeah. So what are you trying to 3D print? The whole ovarian structure, or just the last stage of it? The last, bank part where they're ready to be selected for maturation?
Yeah, that's a good question. So what our first focus is to do the cortical region. So exactly right, the region that the oocytes are banked. And this is because generally we take out one ovary to cryopreserve it and the other ovary remains. And so we wait until that ovary essentially goes through early menopause. We call it ovarian insufficiencies. And then we transplant it back onto that remaining ovary. And so the rest of that tissue is there. Whether or not it's an ideal organ to put it on is another thing that we need to test, because we're sure that a postmenopausal ovary isn't the perfect environment, but the first attempt that we're going to do is to transplant it back onto that remaining ovary. And so we'll just need to make that bank cortical region to house the primordial follicles.
Well, the reason I ask is it's so complicated. Good luck if you could even do a small part of it. So you might as well go for the one part that has the best chance of succeeding.
Yeah, you're really getting at basically our stepwise strategy of our first lines of successes, and then of course there's the there's the ultimate goals of recreating the entire organ, and it functions for a longer time than the natural ovary. That would be ideal. Yeah, for women to not have to go through menopause and then stay that way for about half their life at this point. So that would be awesome if we could recreate the whole ovary.
Well, what happens to the ovaries in menopause? I mean, do they stop growing? I mean, do they still continue apparently as normal? It's just that the oocytes aren't formed at the end, or does the whole thing shut down? Like what's been observed?
Yeah, basically the whole thing shuts down. So there's gonadotropins that control follicular genesis and respond to hormones that come from the ovary. So those are off kilter in response, and the ovary just stops producing estradiol. I mean, it's a decline, so it's not an immediate type thing, though it might feel that way for a lot of women, where they all of a sudden just have all of these symptoms of complete estrogen withdrawal, and no more progesterone. And then there's, of course, other things that the ovary produces. But interestingly, there's still a bank left. Like I said, there's about 1,000 primordial follicles left. It's just not enough for the hypothalamic pituitary gonadal axis of hormones to continue on with that cycle, that would then trigger ovulation, and then result in more estradiol produced. And it's a whole cyclical effect that just kind of stops.
This may be an oversimplification, but I see it as like 3 stages. There's like the bank stage, the primordial part where they hang out. And you said that gets down to be about 1,000, and then they just kind of sit there, I guess, forever. And then there's the growth part in the middle where they grow huge, 600 times the size. And then there's the last stage, now where they're prepped and shoved out the door for conception. So in a postmenopausal woman, the first stage is still there. There's still the bank of cells sitting there, but do they just not progress to the second stage, I call it, where they start to grow and expand and develop? Or is it just exactly. The third stage that's affected?
No, it's the second stage. So they're not recruited to grow, and the growing ones actually produce the estradiol. And then the post ovulatory follicle creates this small gland that produces progesterone and then regresses. So the hormone production happens at what you're identifying as the second and third stages.
Okay,
but
that's good.
Yeah, so the bank of follicles exist from birth to death, but are just kind of hanging out until they're recruited, and then they stop being recruited.
Gotcha. Okay. Yeah, I just wanted to understand it. I appreciate it.
Yeah.
So you're making a scaffolding, so I guess you're modeling that on what the last third or the last part of the ovary looks like, that houses the cells that are just about ready to, again, I just call it head out the door and become candidates for fertilization. That's what you're trying to recreate with your scaffolding, is just that section?
We're actually trying to create the first part, the bank, hoping that they'll be activated within the remaining ovary, and that the scaffold will be dynamic enough, like it was in the mouse model actually, to withstand those changes in size, and actually just be remodeled by the follicles and the other cells that they recruit within the scaffold itself, to actually reform and kind of remake that tissue. So we're kind of assuming that this is what happens in development, and this is what I'm excited to discover in some of our future research studies. But we think that the prepubertal ovary is more like the whole cortex. So once those follicles are triggered to grow, then a medulla type forms, where it gives it the space. And we're hoping to kind of recreate that with the 3D printed scaffold, and with the help of the remaining ovarian tissue, if that makes sense.
So you're saying that science doesn't know the morphology of the ovary and how it changes from birth through puberty to the adult stage? We know what it looks like in adults, but
we know what it looks like in the prepubertal ovary, and we see them all the time, but we don't know how the environment, the microenvironment, the scaffolding proteins, the stromal cells that are around it, how that changes. And that's what we're studying in the lab. But in the mouse model, we were able to create this 3D printed scaffold where it housed the small follicles. But then once they were triggered to grow, because it's made from gelatin, which the cells can interact with and digest and break away, they just reformed an ovary. They recruited vessels when it was transplanted, and it ovulated right through it. So we're hoping that a similar thing will happen when we create it for large animal models and then for humans.
I guess as we don't understand the cell-to-cell signaling within the ovarian structures with the surrounding microenvironment, then with the surrounding other organs and the rest of the organism. So there's like probably multiple levels of signaling going on back and forth. And it sounds like we don't really understand almost any of that yet.
Yeah, and it's really a fascinating part of biology, I think, that if you just give the cells the right environment, that they'll behave how they need to. And that there might be some form of control that we can create within the 3D printed scaffolds, which is exciting, that we might be able to, design something that's even better.
So have you tried different orientations and morphologies of the scaffolding? Given that the body's going to do what it's going to do anyway and remodel the thing, have you found more advantaged structures or morphologies to start out with that seem to work better for you?
Yeah, absolutely, we did. So we, at first, because they're round, thought that we could print these round pores and just kind of slot them in, and that would be good. But it was very hard to actually get the follicles into something that was, meant to be about the same size. And they weren't captured in the pores, like they weren't held within different layers of the scaffolding. Because you imagine we want to fill it to capacity, and there's several layers to this 3D printed structure. It just really didn't work. And so we tried angles then. So we tried 90-degree angles and 60-degree angles and 30-degree angles. And it turned out that 60-degree angles and 30-degree angles were the best ones to support the ovarian follicles. And the 60-degree angle was the one that we actually used for the 3D printed scaffold, because not only was it supportive of the follicle, but it also gave us enough space to put more of them in one piece of a scaffold. So it was quite interesting to kind of go through that and to figure out that we needed to do the angular and advancing offset 3D printing design in order to hold something that was round.
If you're going to 3D print this and make it, why not study how the ovary is formed, in utero when the embryo is developing? I mean, if you can picture it somehow, if you can see it and see how it's developed, maybe it's developed like radially outwards from a central point. Maybe it's built up, I don't know, bilaterally on two sides. It's probably not built up at all like a 3D printed way and layer by layer. Maybe if you're able to figure that out and emulate that, you'd have more success in making it.
Yeah, I see your line of thinking. The problem is the cells of the ovary, and as it's developing, can be singular. So they can be isolated cells, and they can migrate. So they're kind of long and flat in what you would traditionally think of like a fibroblastic type cell. But the oocyte, once we're born, it can't be uncoupled from the granulosa cells. So we need to maintain that sphere. And so it has to be, the scaffolding itself has to be something that has a pocket to hold the sphere together. Does that make sense?
Okay. Yeah, I gotcha. Boy, you've chosen yourself a tough challenge, but it's a good one. That's what I keep thinking. Sheesh. So where are you at specifically with this? So you said it's worked in mice. Are you able to, is a clinical trial in the works for this in people, or is it not at that stage yet where it's working, well enough to do that?
Yeah, it's not at that stage yet. We're testing larger animal models first. And we're doing some investigations, like I mentioned, of what the microenvironment looks like in both larger animal models and some ovarian tissue that that participants donate, which we're extremely grateful for. But we're looking more into the microenvironment because we think it's, humans aren't mice. And so we expect that it will be more complicated than using a gelatin ink for the 3D printed scaffolds, but we'll need other types of proteins and biochemical cues for the follicles for larger animal models. Yeah.
Well, I think just the uniqueness of your gelatin model that allows the cells to migrate and the host to, orchestrate them as it needs to. I mean, I think that's a big innovation. And I'm sure other people that are trying to 3D print and use scaffolds would benefit from that kind of scaffolding themselves.
Yeah, I think so. And especially anything that needs to be maintained in an aggregate structure, the way that our follicles do. I think that this type of design was an important piece to work out. And why not use our favorite, animal model, right? Mice. Plus we were able to use some, genetic manipulations to show that the pups that were born were from the transplant and not from the mom that gave birth to them, if that makes sense. We used a green fluorescent protein, which is always fun. So the pups came out fluorescing green. But yeah, I think that was an important part of the design and to work out the architectural part.
Yeah, definitely. So what animals are next? What animals can you work with?
Yeah, we're thinking of, so we have a few animal models that we're working on for different aspects of this. So we're able to receive porcine and bovine, so pig and cow tissue from slaughterhouses, which is great for us to use because their ovary compartments are more like humans than mice are. So that's what we're using to study kind of the architecture and the design. And then we of course always go back and confirm it with human tissue. But as far as transplants, we'll have to see what type of animals will be the best option for those.
Okay. Well, this is really fascinating.
Thanks.
So what's the best way for people to to learn more? I don't know if they're going to read scientific papers, but how can they look and see what you're doing and learn more, even if they're not like an egg expert?
Yeah. So I definitely have a faculty webpage at both Northwestern University and Lurie Children's. Our clinical website is luriechildrens.org/fertility. And my personal lab website, where you can read little bios of the lab members that work in my lab and hear some updates of what we're doing, is LaRondaLab.org.
Okay, very good. Well, I appreciate you coming. It's been a really cool conversation, and I hope I haven't driven you crazy with the questions.
Oh, no. It's always fun to talk about our research, especially when I feel like I'm informing people about things that they don't normally think about. So thank you.
Oh, yeah.
You're listening to the Future Tech Podcast with Richard Jacobs. Future technologies such as artificial intelligence, stem cells, 3D printing, gene editing, Bitcoin, blockchain, the microbiome, quantum computing, virtual reality, and exploring space are much closer than you might think. In fact, many early versions of these technologies are in play right now, and the companies that are using these technologies are the focus of this podcast. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a thorny medical problem. Remember, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you've enjoyed the podcast, please listen, subscribe, like, and tell your friends about it. Thank you.